Good morning, everybody. It's a pleasure to be back here at the Allen. I was here a couple of years ago, and I mostly talked then about IT infrastructures. This time I'll be talking a little bit more about some of the applications. My talk will be split into basically two components. I'll talk a little bit about the Healthy Brains for Healthy Lives initiative, which if I say Hubble, that's what I mean by Healthy Brains for Healthy Lives. And the second half, I'll talk about connectivity. Louder? Okay. So the starting point of my talk is that we are faced with massive amounts of data in the current era, from subcellular resolution to behavior and everything in between. And we're also swamped with large amounts of data which is available in the public domain. You heard a little bit about that from Danielle just previously with the UK Biobank. There are many other data sets that are also out there. And we are swamped. So in our own context, in Canada, we established the Canada First Research Excellence Fund. This is a federal funding mechanism. And we were funded through that for Hubble, HBHL. And its basic approach and deliverables are to understand the individual brain by combining all of those various components: genetic, epigenetic, physiological, imaging, behavioral, environmental, clinical, and I think importantly as we go forward, social determinants of mental health. This is an area that is, I think, so far very much under-assessed, and I think we have to start operationalizing that information and understanding the individual brain. We need advanced analytics to handle this amount of data. I'll briefly mention that. And our overall goals ultimately are for both clinical and commercialization of clinical translation and commercialization of that basic research. We're all very, I think, quite familiar with the valley of death between basic research and clinical implementation. And by 2022, we have to develop a Canadian framework for brain health, which will essentially be a policy document to guide government in making that leap across the valley of death. The basic Hubble conception is to have four basic themes. A core theme is in neuroinformatics and modeling, and three application domains on the bottom left: models of neurodegenerative disease, on the right cognitive neuroscience, brain plasticity, and population neuroscience and brain health, the underpinnings of both optimized performance and in mental disorders. I speak mostly about Hubble, our own funding, but the CFREF mechanism, Canada First Research Excellence Fund, also provided a substantial grant last year to Evado. The Université de Montréal has a group led by Yoshua Bengio, who's one of the triumvirate of artificial intelligence resurgence, which is giving rise to particularly deep learning. So we are building strong links with that group to establish big data in the brain. We're also collaborating very closely with a sister institute in Ontario at the Western University, which shared basically with us the $150 million from the feds, and we have to work very closely together with common infrastructure. So in total, you're looking at something like $250 million across those three institutions in terms of big data in the brain. The basic rationale for what we're doing is shown by this cartoon, where we are capturing each individual by high-dimensional feature vector, where incorporating any number of behavioral variables, brain phenotype variables, and genetic indices. And this allows us to either explore cohort trajectories through life or individual departures from the normal trajectory, which may be a brain disorder, but it may also be learning or plasticity. It's a change in brain state. And the fundamental premise of the initiative is that all of these application areas can be reduced to a common analytic strategy. Another way of looking at this mechanistic perspective is shown here. Most of the time when we talk about brain disorders, we are implicitly linking gene or gene clusters to a clinical description, a clinical label. And of course, with pleiotropy, you have a lot of entanglement between specific genes and clinical disorders, and it's very hard to untangle those relationships. In fact, what's really going on is better described on the right, where these gene or gene clusters are better associated with a specific mechanism, a specific circuit, a specific function, And those circuits can be found in aberrant form in various clinical disorders. And any one disorder can be viewed as a weighted sum of aberrations of specific circuits. And that circuitry approach is also more amenable to approaches for intervention and manipulation in animal models, and a better understanding of brain disease through a mechanistic deconstruction. An example of this as a general principle is shown here. This is work that we did with, in Alzheimer's disease. We took 733 data sets from the ADNI, Alzheimer's Disease Neuroimaging Initiative. And this shows a beta amyloid distribution across those individuals. We divided the brain up into a set of individual brain regions connected by white matter pathways derived from the Carnegie Mellon White Matter Atlas. And what we're modeling here is the propagation of this beta amyloid from region to region over time, potentially over years, across the entire cohort, and also gives us access to individual metrics. So the bottom right is essentially an exploration of the parameter space for this propagation model. In this case, I stress that this is essentially a glorified compartmental model analysis that is quite familiar to people who know about PET in the audience, positron tomography. So it's a differential equation-mediated propagation model using compartmental analysis. We are actually exploring agent-based propagation models, but that's for another discussion. The broad conclusions of that study are shown here. Essentially, the propagation models, the transfer from any one region to another is characterized by a production or synthesis parameter into the brain, into any given region, and the clearance of that by perhaps the glymphatic clearance system. And what you see here is that there's not much difference between the synthesis parameter across the four clinically defined variables: healthy controls, early MCI, late MCI, and Alzheimer's disease. So there's not much change in the synthesis parameter across those clinical conditions, but there's a very strong change in the clearance term. So the broad message there is that if you're designing a drug for intervention into anti-amyloid therapy intervention for Alzheimer's disease, you're probably not going to get much traction by addressing the synthesis pathway. You really have to look at the clearance pathway. We also examined the relationship between these parameters and ApoE4 status, the major risk factor for Alzheimer's disease. And again, you see not much difference in the production synthesis term based on ApoE4 allele status, but a very strong gene dosing effect on the clearance system. So this is an Alzheimer's disease worked example, but I think you can see that the general procedure can be applied to any other neurodegenerative disorder with a different misfolded protein and a different set of starting brain regions. We're also examining how this might work to understand brain plasticity and development. Time doesn't allow me to go into greater detail, but what I just showed you was the unimodal causal model for beta amyloid propagation. We've also published a multimodal non-causal model where we look at a whole series of brain imaging factors and 200 protein factors to characterize the evolution of pathology in Alzheimer's disease. And we've recently published a multimodal causal model which tries to synthesize between those two earlier models. Hubble is organized like so, as I just described before, main themes. We also have 5 cross-cutting platforms in social science, the NeuroHub, the analytics, clinical knowledge translation, animal models, and cellular and tissue mechanisms. Underpinning the IT infrastructure of the whole enterprise is shown here. This shows you the CBRAIN National High Performance Computing Platform, which binds together 7 HPC sites across the country to give a total of about 200,000 processors. And we get about something approaching 7 to 8 million compute hours on this infrastructure, essentially for free. We just have to write an access application, and that's provided to the country. And this underpins the so-called Canadian Open Neuroscience Platform, which binds together 18 institutions across Canada sharing data. In essence, data can be anywhere, computing can be anywhere, and users can be anywhere. And this is the portal. There are numerous publications on this if you want to go into this in more detail. I spoke on this 2 years ago when I was last here. We're increasingly using cloud services rather than big iron, but the principles are basically the same. The other component of the platform is the Loris database. It started life supporting an NIH study of normal brain development, where we stored specifically imaging and behavioral data across multiple sites in the US. We've more recently added genetics data, and I want to stress that it's not all genetics data. There's no need for us to store all of the raw genetics data. It allows us to store SNPs and CNVs and indels and so on, which then can be used when we're interested in multi-domain imaging, behavioral genetics, indeed epigenetics, and now increasingly tissue sample information. We're doing a lot of work in iPSC manipulation. So Loris allows us to store all of this and conduct joint analysis across multiple sites. Loris underpins two large studies, which I'm going to briefly refer to here. The National Database for Autism Research, NDAR, stores data from the pediatric MRI study of normal brain development across the US, and the IBIS similar study, but this case is looking at autism siblings. I'll give some examples from these two studies now. So NIH PD, as we call it, was started life at the end of 1990s, and we collected data for 10 years for children across 0 to 18 years of age, structural and behavior relationships, and disseminated the results. And many papers have arisen from this very early example of data sharing. One of the behavioral results that came out of this, which I always found it quite entertaining because it's rather counterintuitive. Boys seem to do better in adolescence in terms of the California verbal learning task, which is not language as such. It's a very coarse, simplistic level of word recognition, but still there's a sex differentiation there. On the other hand, girls are significantly better in terms of manual dexterity. This is what is underpinning this. What you're looking at here on the left is it's a clock ticking in months from 0 to 48 months. And just look at the left-hand side for now. What you're looking at here, the growth of myelinated fibers from the central regions, from thalamus down to visual cortex in the first 6 months. And in the second 6 months of life, you see the proliferation through the frontal areas as more integrative aspects of higher brain function start to mature. I can let it go around one more time because you see what it's like at birth. There's not much brain differentiation, and then you see the proliferation of, as I said, the optic radiations and then the frontal areas. Note that this looks rather grainy because this is a composite from 50 brains. It's not an individual brain, it's a composite. This is a 4-dimensional data set in 3 dimensions across time in 50 subjects. These are just 2 other types of MRI image which give us different information, which gives us a better picture. Now that's all very well as a visual story. We can analyze that using mathematical modeling to extract brain shape changes across time in any one subject, and then build maps across all subjects to look at brain growth. And what that is shown here, we look at the volume growth, different views of this, but essentially what it's saying, when you see a bright spot here, it's showing that the cerebellum is growing very rapidly, has grown a lot in 24 months, about 4 times increase in volume over the 48 months that are shown here. And this shows you the growth rate, and basically what they're showing here. The growth rate is very high early on, and then it all slows down as you get to later points to give rise to this 4-fold increase in cerebellum. What this doesn't tell you is that, in fact, parts of the brain are still growing in the later months, but it's been swamped by the large changes early on in brain. So we can take this information And we can normalize it so that we're looking at the relative changes rather than the absolute changes. And that's what I'm going to show you now. What you're looking at here is a change in the absolute brain growth rate. And as you see, it goes to vanishingly small values over the first 24 to 48 months. What's going on down here, as I said, is the relative change. It's not much, but where is it happening most? And it's perhaps not a surprise that the frontal areas are areas which are continuing to grow most at the later stages of the early brain development. So that's ontogeny reflecting phylogeny. We can then extract cortical metrics of brain anatomy. And if we do that, we can get measures of cortical thickness. We can look at brain surface development. This is showing you the changes in the oscillation between mean left hemisphere and mean right hemisphere. It took people like Geschwind and Galaberter basically their entire careers to characterize changes in planum temporale using early MRI sectioning and manual labeling, and now we can essentially do this on 150 brains overnight and characterize the mean changes in brain. What we haven't done yet, and I really do want to do next, is now look at the variation across individuals with respect to this mean, because that's going to capture the information that we really care about in terms of individual variability. We can look at cortical thickness as it changes from birth to 18 years of age, and you see this large area of overgrowth in medial temporal areas, which resolves itself as you go up through the years. We can use this information to look at predictions of chronological age from cortical thickness, and this shows you the relationship between true age and predicted age based on cortical thickness using a general linear model. And we get pretty decent prediction of brain age. But there are some differences, and we can relate those residuals to individual components of performance. And in this case, what we're showing here is the higher the interaction between performance IQ and verbal IQ, the higher the predicted age. So there is an association between the predicted brain age and performance. This has not yet been published. It's in preparation, but this is a mixed effects analysis of 265 of these children in 600 scans to show us that at any given age, the thicker cortex is associated with a higher IQ. I threw this slide in because this is work that was done with our colleagues at the University of Edinburgh studying the Lothian birth cohort. Sharif Karama in our lab worked very closely with Ian Deary and colleagues to establish the relationship between IQ at 70 and IQ at aged 11. And basically what this is telling us is that 2/3 of the variance in IQ can be predicted by let me write. 2/3 of the variance of IQ at age 70 can be predicted by IQ at age 11. So it's not just use it or lose it. A lot of your intelligence at later ages has already been established by the age 11, which is kind of scary. A bit of a diversion for a second here. One of the projects that we're heavily engaged in terms of big data is the so-called BigBrain, which is a cytoarchitectural reconstruction from 7,020 micron sections, each of which is about 50 times smaller than MRI in each dimension. If you do the math, that means that this is equivalent to 125,000 MRIs, or about 1 terabyte of data. And we're using this data. This has been made public quite a while ago in 2013. And last time we looked at it, had been downloaded by 25,000 groups. And we don't know what they're doing with this, which is kind of exciting as an open science concept, but it's also terrifying because we don't know if we're going to be scooped with our own data. But I guess that's what open science means. Lots of people are using it. Just to clarify the point, this is what happens when you zoom into an MRI data set, and this down to, in this case, 2 millimeters. So these are 0.5 millimeter voxels. These are high-resolution MRI voxels. That's what happens when you zoom down to the same area within the big brain. So it's near cellular resolution. We're right now working with our colleagues at Evado on deep learning layer reconstruction. So we get the 6 layers of cortex extracted automatically over the entire brain. One of the things that has come out of this is the definition that layer 4, depth of layer 4 depends on the local curvature, which was something that was posited before, but we've demonstrated it using this fully automated analysis. The second half of my talk is going to cover various aspects of connectivity. Connectivity in an imaging context allows us to look at either direct white matter connections or functional correlations between different brain regions. Indeed, you can even look at gray matter correlations. But this is direct and this is indirect. That allows us to capture information about the entire brain organization rather than looking at any particular focal area. You can develop graph theoretical strategies to quantify the information flow across the networks. Sometimes you look at the shortest path length, this is the Watson-Strogatz characterization. This is also equivalent to what we call global efficiency if you use the Latore and Marchiori approach. The important thing about whether we use global efficiency or local efficiency is that this incorporates direct measurement of brain length and fiber length and network length, and we think that that is absolutely a critical factor in extracting information about these network analyses. So white matter connectivity, one example of that is taken from the IBIS study. This is like the NIH study of normal brain development. This is looking at early brain development in siblings of autistic children, 500 at-risk SIBs. Basically, in autism, a lot of the damage has been done by age 3 when they are clinically diagnosed, and you've missed all of that. But if you look at the siblings of children already diagnosed with autism, then you can basically look at the earliest underpinnings of potential autism, incipient autism. About 15% of siblings will convert to somewhere on the spectrum. So if you do 500 at 15%, it's about 75 of these children we will have followed from essentially birth. We published this just recently. This is showing one of our 6-month-old children. There are 2 papers looking at 24 months in 2014 and 6 to 12 months in 2017, essentially doing the same thing. And that's important because this is where the early action is taking place in terms of behavioral metrics. We know that in autism there is overgrowth in the significant macrocephaly in these early years. This is the result of a meta-analysis from Redke and Korschen back in 2005. And that overgrowth is important because we think that it's that overgrowth which gives rise to long fibers or the loss of long fibers, because the longer fibers, which are the integrative fibers across brain, are the ones which are preferentially pruned out if the brain is too large too soon. So it becomes essentially a question of competition for neurotrophins and energy, and if the long neurons are too long too soon, they get pruned out and they essentially never come back, even if the overall brain size reverts to something like normal. At the end, it's kind of too late. The long-range integrative neurons have been lost. To make the point about length here, you see here that these 2 networks are essentially topologically equivalent. But if you take the 2A regions and flip them from side to side, they have the same topology, but the length between different brain regions is a lot longer. So that's why it's important to incorporate length into the efficiency analysis. So what we see in our children at 24 months is shown here. And to summarize this slide, you see these are the areas where reduced efficiency is associated with ADOS measures of autism severity. And it's largely in early areas. There is a strong association in inferior frontal areas, which are possibly associated with language. But the important take-home message here is it seems that the earliest components of autism seem to be a sensory deficit rather than a higher order deficit. And this suggests that what we see as a clinical manifestation of autism is part of a downstream cascade that arises from these earlier sensory motor deficits. And essentially this says that what I showed you at 24 months is present, although weaker, at 6 to 12 months. I'll finish with a discussion of functional connectivity. Again, it's in the autism context. But here what we do is we look at correlation networks across brain with different seed regions. You can look at the connectivity patterns, and you can start to identify subgroups within the subject similarity grouping, so that we can say that there is not one thing called autism. There are groups which are subgroups which are more similar amongst themselves. And then this allows us to represent any one individual as a weighted sum of those 5 canonical subgroups. So we can start to calibrate our disorder. Rather than just calling it autism, we have, as I said, a set of weights of those 5 subtypes. And I think this is consistent with the mechanistic deconstruction I was talking about at the outset of this talk. In preparing for this talk, I came across this paper from Young Her's group. Young Her was a former postdoc in my lab. He's now a senior scientist in China, and I'm delighted to see how well he's doing, but I thought it was extremely relevant to this meeting. What his group has just done is to examine the Chronnectome, which is the temporal dynamic changes in the functional connectome. The whole premise here is that it's those dynamics which are really important to characterizing the individual. And they established a stable and remarkable individual variability in indices of brain dynamics, whether they use strength, stability, or variability of the dynamic functional connectivity matrix. They found spatial patterns identified individuals with high accuracy. I'm going to explain this with a couple of slides in a moment. And significantly predicted individual cognitive performance, particularly fluid intelligence and executive function. These individual differences were primarily mediated by higher order networks, the default mode network, the dorsal attention network, and the frontal parietal network. And by capturing individual brain network dynamics, this has implications for individualized characterization of health and disease. So how is this being done? You essentially develop a regional atlas. You can divide it up into 250 brain regions in this case. So we can look at the correlation across time between any 2 brain regions. If you have a moving time window, you can extract that information for blocks of time during the entire session. And this allows you to build up a temporally changing functional connectivity map. And you can extract various metrics of that across subjects. What becomes very interesting here is that this allows them to compare people in session 1 and session 2, and can they be uniquely identified on the basis of these metrics. And they found very high correlation between session 1 and session 2 for these 3 variables, particularly strength and variability. So this allows, if you have that measure of specificity, about 95 out of 105 subjects were correctly identified from session 1 to session 2, or from session 2 to session 1. So this means that those functional variations in individuals are real and reproducible for the individual. And these can be, you can have a strong correlation between the predicted and observed fluid intelligence, predicted and observed cognitive flexibility, and predicted and observed executive inhibition. You can even look at where specific subnetworks, for instance, the default mode network, brain regions which are in the default mode network have a strong influencing effect on the prediction for cognitive flexibility. So we can start to parse down into finer detail which parts of brain are involved in individual variability. This is work that's very preliminary from my lab, so I don't want to talk about it too much. I just wanted to throw it in at the end to note that this functional connectivity is heritable, and we can identify subregions which have a significant heritability across the population. And this is something that I think we will be exploring more in future detail. So that's all I want to say, unless people have questions about the IT infrastructure. I've got a lot of slides, but I'm also about to run out of time, I believe. So, so I'm going to stop there. Thank you very much.
Okay. Are there any questions? Back in. Yep.
Just in respect to the autism part of the talk, where you divided the autistic subjects up into these various groups, did you try to relate that in any way to the genetic heterogeneity of autism?
We haven't done that with this data set. I think at the end when I talked about the heritability of functional connectivity, which is what we're doing right now, that's an obvious. I showed you one result for normal brain, but applying that same machinery to the autism population is what we'll be doing next.
I think you said something about being able to predict IQ, say as measured on a paper and pencil test from cortical thickness. Could you just comment on what the current status is in terms of the biophysical measurables that can be directly correlated to IQ?
I think I said that we were showing the prediction of brain age, from cortical thickness. So that was the Kundrak-Pam paper. So you can get a good prediction of brain age. It's the residuals that we saw there that seem to be associated with the interaction between performance IQ and verbal IQ. So what is the biological substrate of that? I showed a slide where we showed the cortical thickness seems to be thicker for higher IQ across the entire age range from that mixed effects analysis model, which has implications because cortical thickness, as measured from MRI, is essentially a bulk parameter. It doesn't tell us what is going on inside that cortical area. You are essentially getting the boundary between gray matter and white matter as visible on MRI. So that change in contrast can arise from increased dendritic arborization, myelination, increases in neuropil. They can all give rise to microstructural changes in cortical structure. And with non-invasive MRI, we are not in a position to get down to the microstructure over the whole brain. We are hoping that we can start to use things like 7 Tesla MRI to get down to finer resolution, perhaps down to 200 microns. And then we can use layer information from the big brain as a prior to help us interpret changes in cortical layer structure within in vivo non-invasive MRI. That's the next point on the horizon for us. But the reality is that MRI today has a resolution of typically 1 millimeter, 1,000 microns, and at best 500 microns, which is far above cellular level.
Okay, thank you so much.
Thank you very much.